So the goal of this lecture is really to talk about how insulin is stimulated to be released by pancreatic beta cells and how then glucose is uptaken by our muscle cells. To start off with, though, we are going to talk about carbohydrates. Carbohydrate is synonymous with the term sugar. Okay? So carbohydrate, sugar means the same thing. We have three classes of carbohydrates or sugars. We have monosaccharides, we have disaccharides, and we have polysaccharides. Monosaccharides, disaccharides, and polysaccharides are all types of carbohydrates or sugars, but structurally, they're a little different from one another. And they're also very similar. So let me explain. Monosaccharides are the simplest carbohydrate or sugar. Monosaccharides are singular units of either galactose, fructose, or glucose. Again, monosaccharides are a type of carbohydrate or sugar, and to be a monosaccharide, it needs to be a singular unit of either galactose, fructose, sometimes called fructose, and glucose. So when and where do we find singular units of galactose, fructose, or glucose? An example would be like in honey, we find glucose and fructose. In berries, so different types of fruit, we'll find glucose and fructose by themselves. Those are examples of where you would actually find monosaccharides in nature. All right, so what's another type of carbohydrate or sugar? Disaccharides. Disaccharides, it says it kind of in the name, disaccharides are going to be two monosaccharides bound together. So here's one that everybody knows, sucrose or table sugar. What is sucrose or table sugar? It is a glucose monosaccharide and fructose monosaccharide bound together. Anything that you want to taste sweet, you can put sucrose into. It's in a magnitude of things. Drinks, cereal, it's in cakes, it's in bread. We add sucrose or table sugar to all kinds of stuff. All right, so what's another disaccharide? Lactose. Lactose is milk sugar. So this is the sugar that we find in milk. And what lactose is made up of is a glucose monosaccharide bound to a galactose monosaccharide. This is what makes milk taste sweet. Generally speaking, although this term is by no means truly scientific, we consider monosaccharides and disaccharides to be simple sugars. You could also use the term simple carbohydrate because sugar and carbohydrate mean the same thing. Why are these carbohydrates or sugars called simple? Because monosaccharides are literally singular units of either galactose, fructose, or glucose, and disaccharides are two only monosaccharides bound together. So neither of these are very complex, which brings us to the conversation of what's a complex carbohydrate. And typically we would consider complex carbohydrates to be polysaccharides. So what are polysaccharides? Typically polysaccharides are 10 or more monosaccharides bound together. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, many more than 10 monosaccharides bound together here. This is showing you glycogen, which is the storage form of glucose in the human body, although in plants that would be starch and it would really look pretty much identical. What we would see is 10 or more glucose monosaccharides bound together. And since there's 10 or more glucose monosaccharides all bound together, we would refer to polysaccharides as being complex. Starch, again, is a great example. We find starch in plants. An example of polysaccharide starch in plants, we would find polysaccharides in grains that we use for, making bread, quinoa, oats, sweet potatoes. We find polysaccharides, specifically starch, in lots of different plants. Again, it's the storage form of glucose in plants, and we consume it. Cellulose, for example, is another polysaccharide. However, Cellulose, which is again located in plants as well, and is a polysaccharide, is insoluble. So we refer to it actually as a fiber. But this is not a nutrition class, so we're not going to get into all of that. The main polysaccharide that we consume in nature, and ultimately to utilize in our cells to make ATP through metabolism, oxidative phosphorylation, is going to be starch. All right, so most of us like to eat carbohydrates, be it monosaccharides, disaccharides, or polysaccharides. Most of us eat a combination of those three different types of sugars or carbohydrates. And when we eat them, right, they go into our mouth, and they go down into our esophagus, then they enter into our stomach, and then they enter into our small intestines. And within the small intestines is where the majority of carbohydrates are actually going to get absorbed. So let's talk about that. The small intestine can be divided into three different sections. First of all, you have the duodenum, sometimes called the duodenum, either is fine. Then we have the jejunum, then we have the ileum. Okay, I'm telling you that because I'm going to go over some blood vessels real quick. Here you can see the mesenteric artery, and the mesenteric artery carries oxygenated blood, and what the mesenteric artery is going to branch into the pancreatic duodenal artery. Also could be called the pancreatic duodenal artery. That's going to carry oxygenated blood to the duodenum or duodenum. And we can also see that the mesenteric artery is going to branch into the jejunal arteries, which is going to carry oxygenated blood to the jejunum. And then we also see that the mesenteric artery branches into the ileal arteries, which is carrying oxygenated blood to the ileum, the different portions of the small intestine. So now what we're doing is looking at veins carrying deoxygenated blood. So here you can see the pancreatic duodenal or duodenal veins carrying deoxygenated blood back from the duodenum or duodenum. Here you can see the jejunal veins carrying deoxygenated blood back from the jejunum. Here you can see the ileal veins carrying deoxygenated blood back from the ileum, and they are all going to merge onto and form the mesenteric vein, which is going to carry the deoxygenated blood ultimately to the liver. So what's the point of showing you this sort of vascular anatomy? The different arteries and veins that supply the small intestines. Well, the reason for that is because, well, as the carbohydrates travel through the small intestines, what's going to happen is those carbohydrates, those sugars, are going to get absorbed past the small intestinal wall and ultimately into these different veins. Right? They're ultimately those carbohydrates, well, first into the capillaries, but they're going to get absorbed past the small intestinal wall, and those carbohydrates are going to end up in the pancreatic duodenal veins, in the jejunal veins, and in the ileal veins. And now we have carbohydrates or sugars in the bloodstream. We have to get those carbohydrates or those sugars past the wall of the small intestines and into these different veins that supply the small intestines in order for what we call blood sugar, blood carbohydrate, whatever you want to call it, to rise. Right? That's how blood sugar is going to increase, because once that carbohydrate or sugar is past the small intestinal wall and into these veins, what's in the veins is blood, right? Blood sugar is now going to be elevated. So what I want to talk with you about is, of course, how that absorption occurs. And I have to mention the mesenteric vein, before it brings the deoxygenated blood to the liver, it becomes what's known as the portal vein or hepatic portal vein. I mean, that would be a huge issue anatomically if I didn't say that. Not necessarily that matters for this class, but the hepatic portal vein is what brings the blood ultimately to the liver, not the mesenteric vein. The hepatic portal vein is ultimately going to be formed from the mesenteric vein, but they're not the same thing. So here is the lumen, the hollow opening of the hollow opening in the center of the small intestines. What's traveling through the lumen? Carbohydrates, monosaccharides, disaccharides, polysaccharides, right? All those di and polysaccharides are going to get turned into monosaccharides, which we'll talk about in a minute. But ultimately, what's going to happen is these different carbohydrates or sugars are going to have to get absorbed past the wall of the small intestine so they can go into the bloodstream. So what does the wall of the small intestines look like? What's it made up of? Over here, you can see the wall of the small intestines. We're not going to acknowledge for now, because this is really more of an anatomy conversation, that the wall of the small intestines is organized into these finger-like projections called villi. That's not important. Don't let that confuse you. What I want you to see here is the wall of the small intestines is lined with these columnar epithelial cells. The wall of the small intestines, for those of you that really like anatomy, is lined with simple columnar epithelium. So what we need to do is we need to get the glucose, which is within the lumen. This is where the lumen would be. We need to get the carbohydrates, the sugars, past the wall of the small intestines. Okay? The wall of the small intestines is made up of these column-shaped epithelial cells. So we need to get the carbohydrates past these column-shaped epithelial cells. And once these carbohydrates get past these column-shaped epithelial cells, then they can enter in to the capillaries, the blood capillaries. Okay, where are these capillaries coming from? Let's pretend that this portion of the small intestines is actually a portion of the duodenum. Okay, so what you would have here would be the duodenal or duodenal arteries, and the duodenal or duodenal arteries would become ultimately the capillaries. You can see the capillaries here. This is getting a little messy, but you can see the capillaries here lying behind the small intestinal wall. So if we can get these carbohydrates past the small intestinal wall, past these column-shaped epithelial cells, then the carbohydrates can enter in to what would be the equivalent of, in this image, if what I'm saying is accurate, we can get those carbohydrates to enter into these duodenal capillaries. Great. So now, what's within the duodenal capillaries? Well, of course, blood. So what's within the capillaries? So blood sugar is going to rise. And these duodenal capillaries will ultimately become over here the duodenal veins, which will become the mesenteric veins and hepatic portal vein, and the blood will be taken to the liver. That's not really the important point, though it's not bad to know that. The important point is what I'm trying to get across here is if we can get these carbohydrates across the wall of the small intestines, across these column-shaped epithelial cells, then the carbohydrates can enter into the bloodstream, enter into the duodenal capillaries in this example, and blood sugar will rise. All right, so over here is the lumen. Okay, these are the epithelial cells that line the small intestines, and over here are going to be where the blood capillaries are. Okay, so what do we need to do? We need to get the carbohydrates that are within the lumen, ultimately, past these column-shaped epithelial cells so that they can enter into the capillaries and cause blood sugar to rise. Okay? So how are we going to get these carbohydrates past these epithelial cells that line the small intestines? That is the question. First of all of the di and polysaccharides that you consumed within the small intestines through enzymes like amylase enzymes and all sorts of different enzymes are going to work to break those di and polysaccharides down into individual monosaccharides. Okay, so sucrose, for example, will get split into glucose and fructose monosaccharides. And if it's a polysaccharide, then what's going to happen in the small intestines? This happens even at the beginning in the mouth, for example. There are amylase enzymes within our saliva. But regardless, especially by the time we're in the small intestines, the polysaccharides that are made up of 10 plus glucose monosaccharides, the polysaccharides are all going to get split into individual monosaccharides as well. So what you're going to have in the lumen of the small intestines is a whole bunch of monosaccharides. because the disaccharides and polysaccharides that you consumed are all going to end up being monosaccharides by the time that they are within the small intestines. Unless, of course, the polysaccharide that you consumed is something like cellulose, which is not soluble. I'm talking about starch. If you eat starch, polysaccharide starch from plants, by the time starch, the polysaccharide from plants, ends up in the lumen of the small intestines, all the individual glucose monosaccharides are going to have been broken off. So how do we get all of these monosaccharides within the lumen of the small intestines now, past these epithelial cells and into the bloodstream? To start off with, it's going to be, well, pretty easy. Glucose levels are going to be quite high in the lumen compared to inside of the epithelial cell. Okay, so the portion of the epithelial cell that faces the lumen, the hollow opening, is going to be called the apical side of the epithelial cell. Any cell that faces an opening, that side is always called the apical side. So what we need to first do is get glucose, which is in the lumen, past the apical side of the epithelial cell and into the epithelial cell. And we're going to move glucose from a high concentration to a low concentration inside of the epithelial cell. So how are we going to accomplish that? Well, first of all, initially we're obviously going to use passive transport. We're going to move glucose from a high concentration to a low concentration. We're not going to need energy in order to do this. So what we find located in the apical membrane of the epithelial cell are going to be integral proteins which function through facilitated diffusion. This is what's called GLUT2, which is a glucose transporter, sometimes called GLUT2, that's located in the apical membrane of the epithelial cell. That's obviously the apical membrane of the epithelial cell is a plasma membrane because epithelial cells are cells, right? And what we find is an integral protein that functions through facilitated diffusion. So initially what's going to occur, this GLUT2 protein, integral protein, is going to be open facing the lumen and closed off facing the intracellular fluid. And what glucose can do, or these other monosaccharides that exist within there, is enter and dock into the GLUT2 protein. And what the GLUT2 protein will do is then it will open up facing the intracellular fluid of the epithelial cell and close off facing the lumen. And that will allow glucose in to the epithelial cell. So we just got glucose into the epithelial cells that line the small intestines through facilitated diffusion. And now we've got glucose inside of the epithelial cell. And what we need to do is now we need to get glucose past what's called the basolateral membrane of the epithelial cell so that it can enter into what's called the extracellular or interstitial fluid and then enter into the bloodstream, get into the capillary. Let's be specific about anatomical terms now. Extracellular fluid is technically the fluid that exists outside of our cells plus the fluid inside of our vessels, so the blood. What I'm really saying here is we need to get glucose inside of our epithelial cells that line the small intestines past the basolateral membrane of the epithelial cell and into what's called the interstitial fluid. The interstitial fluid is the fluid that exists right outside of our cells and does not include the blood. Okay, extracellular fluid as a whole is the interstitial fluid plus the blood within our vessels combined together. If you're just talking about the fluid that exists directly outside of the cells that's not inside of the vessels, then it's interstitial fluid. So what we really need to do is we need to get glucose into the interstitial fluid, which is the fluid that lies just outside of the basolateral membrane of the epithelial cell. So how are we going to accomplish that? Well, comparatively, glucose is going to be higher inside of the epithelial cell than it's going to be inside of the interstitial fluid. Because the glucose that ends up in the interstitial fluid through a variety of different Starling forces that we'll talk about later on in the course, that glucose is constantly getting sucked into the blood capillaries and being carried away. So blood glucose levels stay very low in the interstitial fluid outside of these epithelial cells. So again, we're moving glucose, relatively speaking, from a higher concentration inside of the epithelial cell past the basolateral membrane into a lower concentration of glucose in the interstitial fluid. Okay, so how are we going to do this? We're going to do it through facilitated diffusion, again, located in the basolateral membrane of these epithelial cells are more GLUT2 proteins, okay? Integral proteins that function through facilitated diffusion. And initially, they're going to be open, facing the intracellular fluid of the epithelial cell, and closed off, facing the intracellular fluid. So glucose can come in, whatever monosaccharide this is, and it can dock into the GLUT2 protein. And what the GLUT2 protein then will do is it will close off, facing the intracellular fluid, and open up, facing the interstitial fluid, and glucose will then move into the interstitial fluid, and then it can be carried into the blood within the capillaries. So to start off with, right after you've eaten some carbohydrates, you actually get those carbohydrates from the lumen of the small intestines, past first the apical membrane of the column-shaped epithelial cells, and then the basolateral membrane of these columnar epithelial cells through facilitated diffusion on both sides, right? Because relatively speaking, you're moving, let's just say this one more time, glucose, that was a bad drawing of glucose. You're moving glucose from a high concentration in the lumen into, relatively speaking, a low concentration in the epithelial cell. So high concentration in the lumen, low concentration in the epithelial cell, we can use facilitated diffusion. And then again, we're going to move glucose from the epithelial cell, now past the basolateral membrane, into the interstitial fluid. And again, we're moving glucose inside of the epithelial cell to the interstitial fluid, where glucose is relatively high. Again, speaking specifically about glucose in the epithelial cell versus the interstitial fluid, glucose is high in the epithelial cell and relatively low inside of the interstitial fluid. So we're moving glucose from high to low again, past the basolateral membrane. So we can do it again through facilitated diffusion using GLUT or GLUT2 proteins. We are eventually going to run into a problem here, though, because what's going to happen over time is once a lot of this glucose gets absorbed, right, eventually the glucose levels within the small intestines are going to be relatively low. And the glucose concentrations inside of the epithelial cells is going to be relatively high. Again, this is going to begin to occur, especially at the end of absorption of glucose. And a healthy human being does not allow any glucose to remain in the small intestines. Our bodies are very good at ensuring that the vast, if not all, glucose within the small intestines actually gets absorbed past the small intestinal wall, these epithelial cells, and into the bloodstream. So the question is, how are we going to accomplish that? Because we're not going to be able to use facilitated diffusion anymore, right? Because, well, relatively speaking, glucose is lower in the small intestinal lumen than it is within the epithelial cells that line the small intestine. So what are we going to do? Here you can see another depiction, just a little different. This is the lumen. This is the epithelial cell lining the small intestines. And over here is where the interstitial fluid and capillaries are located. Okay. So again, same thing applies. We need to get the monosaccharides, I keep saying glucose, but all the monosaccharides within the small intestines, we need to get them past these epithelial cells and into the interstitial fluid so they can enter into the bloodstream. Okay, but now I'm talking to you about a circumstance where glucose, this is at the end of absorption, where glucose is actually lower in the small intestines. small intestinal lumen than it is within the epithelial cells that line the small intestines. So if we want to move glucose from a lower concentration in the lumen past the apical border of the epithelial cell into the epithelial cell where glucose is relatively high, how are we going to accomplish that? Well, we're going to have to use some type of active transport because now we're trying to move glucose from a low concentration in the lumen into a high concentration in the epithelial cell. So GLUT2 proteins are not the only proteins that exist, integral proteins, transport proteins that exist in the apical membrane. We also have active transporters located in that membrane as well. Specifically, the type of active transporter, integral protein, that we have embedded in the apical membrane of the epithelial cell is one that functions through secondary active transport. So what I can tell you is sodium levels are always going to be maintained at a higher level inside of the lumen than they are inside of the epithelial cell. Why? Well, we haven't talked about this yet, but sodium is constantly through active transport getting kicked out of the epithelial cell and into the interstitial fluid. So sodium levels always stay, like all the cells in our body for the most part, sodium levels stay very, very low inside of these epithelial cells that line the small intestines. Okay, so sodium is going to have a driving force, a diffusion gradient, to move from the lumen into the epithelial cell past the apical membrane. And when it does, what occurs is it passively diffuses by an integral protein. And it provides that integral protein the energy that it needs to move glucose and other monosaccharides in the lumen from a relatively low concentration in the lumen to a higher concentration in the epithelial cell. Again, this is happening at the end of absorption when glucose levels are actually lower in the lumen than they are in the epithelial cell. Okay, so we need to move glucose from a low to a high concentration past the apical membrane. And we're going to, of course, need active transport to do that. So what type of active transport is utilized? Secondary active transport. The diffusion, let me erase this a little bit, the diffusion of sodium ions into the epithelial cell, right, from the lumen into the epithelial cell, provides this integral protein the energy that it needs to move glucose from the lower concentration in the lumen to a higher concentration inside of the epithelial cell. That's how we get glucose past the apical membrane, even when glucose levels are lower in the lumen than they are in the epithelial cell. And then the second part of this is easy. What we're going to be doing still is we're going to now need to move glucose from a higher concentration in the epithelial cell to a relatively lower concentration in the interstitial fluid. Why is it still lower? Well, again, as the blood travels through the capillaries through forces that we haven't discussed yet, glucose is constantly getting pulled into these capillaries, so glucose levels stay very low in the interstitial fluid. Okay, so now we're going to move glucose from a higher concentration in the epithelial cell past the basolateral membrane to a lower concentration in the interstitial fluid. So we need to move glucose from a high concentration to a low concentration so we can use passive transport. Specifically, we're going to use more of these GLUT2 proteins. The same integral proteins are going to get used through facilitated diffusion. The GLUT2 proteins are going to be open up facing the intracellular fluid and closed off facing the interstitial fluid, allowing glucose to come in and dock. And then what will happen is they will change their orientation so they're closed off facing the intracellular fluid and opened up facing the interstitial fluid, and glucose will be able to exit out into the interstitial fluid and then enter into the bloodstream. Okay, so let's briefly recap here. At the beginning of absorption, glucose is higher in the lumen than it is inside of the epithelial cell, so it can travel past the apical membrane through facilitated diffusion using a GLUT2 protein. Glucose still is going to be relatively high in the epithelial cell compared to the interstitial fluid, so it again can move past the basolateral membrane through a GLUT protein, facilitated diffusion again. Okay, this is what happens, the process by which glucose gets from the lumen into the interstitial fluid and into the bloodstream at the beginning of absorption, after you've eaten. Then we will reach a point, especially towards the end of absorption, where glucose is actually lower in the lumen than it is within the epithelial cell. So now, in order to get glucose from the lumen into the epithelial cell past the apical membrane, we're going to have to use active transport. And what gets used here at the end of absorption is secondary active transport using sodium. And that's what actually allows us to get glucose from the lumen into the epithelial cell from a low to a high concentration. And then again, this ultimately ends up being the same thing as the last example. Glucose within, I keep circling fructose, but glucose within the epithelial cell is still, in this example, going to be higher than it is within the interstitial fluid, which is constantly getting pulled into the blood within the capillaries. So again, we're just going to move glucose from a high concentration in the epithelial cell to a low concentration in the interstitial fluid. So to get it past the basolateral membrane, we can still just use those GLUT2 proteins through facilitated diffusion. So these mechanisms that I've discussed with you ensure that the vast majority, if not all, of the carbohydrate that we eat ultimately gets from the lumen of our small intestines past the wall of our small intestines, our epithelial cells that line the small intestines, into the interstitial fluid and into the bloodstream. Okay. There are other proposed mechanisms as well that aren't, I would say, as popular. We could spend the entire semester talking about them, but I'm going to give you the ones that are, I would say most well accepted. Okay, so what am I getting at? After you eat carbohydrates, be it monosaccharides, disaccharides, polysaccharides, the vast majority of those carbohydrates, once they become monosaccharides within your small intestines, are going to get absorbed into your bloodstream. And your blood sugar levels are going to rise, right? Quite a bit. The monosaccharides, first of all, if you actually eat monosaccharides and disaccharides, and this conversation gets complicated by fiber and all kinds of other stuff, but again, it's not a nutrition class. Let's just say this. Monosaccharides and disaccharides, because of their simple nature, tend to get absorbed faster and cause blood sugar levels to rise. Polysaccharides take time to break down and thus take longer to absorb, so they cause blood sugar levels to rise at a slower rate, and thus that's why they're referred to oftentimes as being healthier. But at the end of the day, regardless of the type of carbohydrate that you eat, the vast majority of it is going to end up within your bloodstream, and your blood sugar levels are going to rise. So now the conversation shifts to what happens when blood sugar levels rise. When blood sugar levels rise, that is going to influence your pancreas. Okay, within your pancreas, there are beta cells highlighted in red here. Okay, and what the beta cells do is they produce insulin. Okay, and when blood sugar levels rise, what they're going to do, what an increase in blood sugar does, is it's going to trigger these pancreatic beta cells to secrete insulin. And of course, that's simple to say. But what about blood sugar levels rising causes pancreatic beta cells to actually secrete insulin? That is a much more sophisticated question that requires a knowledge of many of the things that we have discussed so far in this course. So here we go. All right. First of all, this is a pancreatic beta cell. Okay, this cell here, that's a beta cell. Located in the membrane, plasma membrane of our beta cells are GLUT2 integral proteins. So what does that mean? Well, if there's more glucose in the bloodstream than there is inside of the pancreatic beta cell, well, then glucose is going to move from a high concentration in the blood to a low concentration in the pancreatic beta cell through facilitated diffusion, right? This integral protein is going to be open up facing the extracellular or interstitial fluid in this example, and closed off facing the intracellular fluid, and glucose will move in to the GLUT2 protein, and it will close off facing the extracellular or interstitial fluid and open up facing the intracellular fluid, and then glucose will enter. So now we're actually, as blood sugar levels rise, glucose will actually begin to enter into your pancreatic beta cells through facilitated diffusion. This glucose that enters in will then be ran through glycolysis, as well as, we haven't talked about this yet, but pyruvate will enter into the mitochondria and be used in oxidative phosphorylation. At the end of the day, glucose metabolism will begin, and we will start to produce more ATP inside of the pancreatic beta cells, right? Because glucose can be used in order to make ATP. Glucose and fatty acids are the two primary macronutrients that are used inside of our cells for the production of ATP. Okay, so if more glucose is entering in through the pancreatic beta cell and more of the glucose is being metabolized, then our ATP levels within the pancreatic beta cells will begin to increase. It looks like what ATP does is ATP goes over. When ATP levels are elevated, what ATP does is it actually inhibits or closes potassium channels in the plasma membrane of these pancreatic beta cells. It causes them to shut, right? These are like potassium leak channels, essentially, and they're typically open. And when ATP levels are rising inside of the pancreatic beta cells due to increased glucose metabolism, these potassium leak channels are actually inhibited by elevated ATP levels, and they begin to shut. And now this brings us back to membrane potentials. Pancreatic beta cells have resting membrane potentials of about -80 millivolts. Okay, great. That tells us something about the number of potassium versus sodium leak channels, right? There's many more potassium leak channels than there are sodium leak channels, right? Eventually we'll get to a point where potassium leaves at the same rate that sodium comes in. Great. Okay, so think about this. What would happen if you were to close or shut some of the potassium leak channels? Okay, you would reduce the rate at which potassium, of course, would be able to leave, right? Because you'd be shutting off these potassium leak channels. You'd be reducing the rate that potassium can leave, but you wouldn't be reducing the rate, for example, that sodium could come in, because those leak channels are still open. Okay, so when ATP levels rise, they close some of these potassium leak channels and cause them to shut, but that has no influence over the sodium leak channels. Okay, so what are you doing? You're slowing down the rate at which potassium can leave, but allowing the rate at which sodium comes in to stay the same. So what that's going to translate into is relatively simple. Now sodium is going to be entering into the cell at a faster rate than potassium is going to be leaving. If you close the potassium leak channels and give potassium less doors to leave through, the rate at which potassium will be able to leave will decrease. And comparatively, now sodium will be entering into the cell at a greater rate than potassium is leaving. So the cell is going to be gaining more positively charged ions than it's losing. So what's going to begin to happen to the inside? It's going to begin to depolarize, is what we say. It's going to begin to become more positive. It's going to begin to go from -80 to -70 to -60, so forth and so on, right? The pancreatic beta cells are now actually going to begin to depolarize. They're going to begin to become more positive. All right, what exists in the plasma membrane of pancreatic beta cells are what are called voltage-gated calcium channels. These are calcium channels that are closed. Okay, they're gated. Okay? They're not open, right? But they can be opened by a voltage. Basically, if you depolarize, make the interior of the pancreatic beta cells more positive, which is exactly what we're doing right now, right? We're depolarizing the pancreatic beta cell. What that will do is it will cause these voltage-gated calcium channels, right? They're voltage-gated. It'll cause these voltage-gated calcium channels to open up because the voltage on the inside of the pancreatic beta cell is changing. It's becoming more positive, and that causes the gates located on these voltage-gated calcium channels to open. And that then allows calcium to move from a high concentration in the interstitial fluid to a very low concentration in the intracellular fluid. So now there's an influx of calcium ions into the pancreatic beta cell. Okay? So intracellular calcium levels inside of the pancreatic beta cells are rising. So what the calcium does that just entered in through the voltage-gated calcium channels, right? Calcium levels are rising in the intracellular fluid. This calcium that comes in causes what's known as calcium-induced calcium release. Inside of the pancreatic beta cells, there is, just like most of the cells in our body, there's the endoplasmic reticulum. The endoplasmic reticulum does a lot of things, but one of the things that it does is it stores calcium. Okay, and that influx of calcium from the interstitial fluid into the intracellular fluid of the pancreatic beta cell, what it does is that calcium goes over and it binds to channels located on the endoplasmic reticulum inside of the pancreatic beta cells. And these channels are called RYR channels, or ryanodine receptors. And it causes these ryanodine receptors or channels to open up. And what happens is when these ryanodine receptors, these channels open up, is calcium is able to leave the endoplasmic reticulum into the intracellular fluid of the pancreatic beta cell. So again, what's really accomplished here through this influx of calcium through these voltage-gated calcium channels due to depolarization of the pancreatic beta cell is calcium-induced calcium release of the endoplasmic reticulum. The calcium within the cell binds to channels on the endoplasmic reticulum and allows the calcium inside of the endoplasmic reticulum to travel out into the intracellular fluid. So collectively, we've now got a whole bunch of calcium ions elevated inside of the pancreatic beta cell. And what are all these calcium ions going to do? Located inside of the pancreatic beta cells are vesicles filled with insulin, okay, the hormone. And what happens is when calcium levels elevate inside of the pancreatic beta cells for all the reasons that we just discussed, what calcium does is it triggers the vesicles to go and dock to the plasma membrane of the pancreatic beta cells and allow the insulin out via exocytosis. So that elevation in calcium within the pancreatic beta cell causes the exocytosis of insulin, and that insulin then ends up in the interstitial fluid and enters into the bloodstream that lies around the pancreatic beta cells. So what's really happened here is the pancreatic beta cells have released insulin that's now in the bloodstream. Right? The pancreatic beta cells have released the hormone insulin into the bloodstream through this quite sophisticated mechanism that we just went through. Okay? Without rehashing everything that I just went through, elevations, and you're taught this in the most basic level. You said, when blood sugar levels rise, the insulin, the pancreas releases insulin. But this is the mechanism by which elevated levels of blood sugar causes pancreatic beta cells to actually release insulin into the blood. And in the next lecture, what we're going to do is talk about how insulin levels rising in the bloodstream, right? When blood sugar levels increase, pancreatic beta cells release insulin, and now insulin is elevated within the bloodstream. We're going to talk about how insulin rising within the bloodstream actually goes over to our skeletal muscle cells and allows our skeletal muscle cells, imagine this as the plasma membrane of a skeletal muscle cell, how insulin triggers the skeletal muscle cells to be able to take up that glucose that is now elevated within the bloodstream.